Greetings, everyone. Welcome to our 2:30 press conference, which is about cholera in a time of changing climate. We intended to have 4 speakers, but various circumstances have made it difficult for those speakers to be here. We're hoping we have a conference call open for them. We've informed them that they can call into the press conference if they are able to. So we might get voices appearing out of the ether at any minute who will join us and give their talks remotely. But in the meantime, we're going to go ahead with our 2 speakers who are here and see how it works. So our speakers who are going to talk in this order are Ali Akanda. He's from the University of Rhode Island in Kingston, Rhode Island, and Antarpreet Jutia. He's from West Virginia University in Morgantown, West Virginia.
We have a presentation up there. There's only one. This one. It's in Excel. I think it's not open, but that's okay. Yeah. Yes. Yeah. Okay. Start trying. Sure. Good afternoon, everyone. Sorry. So we are talking about this very interesting topic, cholera in a time of changing climate. And unfortunately, Dr. Caldwell is not here yet, but we will try to convey as much information as we can. And so we're just setting the stage that the global it's like diarrheal diseases are currently having a tremendous toll on public health, and cholera is a signature disease among these, which is re-emerging in Africa over the last 10 or 15 years. And as you can see from the map that the last 10, 15 years have seen increased outbreaks in almost all over coastal Africa. And we want to understand how we can use this how changing climates are going to impact these progression of the disease. On the right side of the map, you see the subcontinent, the Indian subcontinent, where cholera is understood to have started from, which is we call the native homeland. And we want to use our understanding of the cholera, the endemic cholera in this region, to understand how it is progressing in the African region, where it is on clearly on a march. This figure shows clearly that the trend in Africa is simply devastating, and over the last 20 to 30 years, cholera has increased significantly in almost all parts of Africa. We see here the total count in the shown in the red, and the 3 different regions: West Africa, Southern Africa, and Central Africa. So primarily Sub-Saharan Africa, but divided into 3 regions. But all 3 regions show a very strongly increasing trend in cholera cases that is reported to the WHO, the World Health Organization. And our goal is to use the understanding of cholera and climate relationships seen in the endemic areas to better predict cholera in this African region. So if we want to see cholera in the native homeland, so the Ganges and the Brahmaputra river basins, which have formed the Bengal Delta, is the key centerpiece of the understanding of cholera and climate and hydrology relationships. And this is a picture that we see in where these rivers come together in Bangladesh, and we see a clear combination of endemic and epidemic cholera. We see 2 peaks, one in spring and one in fall, and we see endemic peaks in spring in the coastal areas and also in the central areas, and we see epidemic peaks in inland and also parts of the central areas. And it is mainly driven by this highly seasonal hydrology that these two rivers show, and this is a clearly asymmetric picture of water availability that drives these progression of cholera, and it's also highly variable interannually. Both rivers, they have very strong interannual variability that creates the water-related natural hazards that impacts the progression of this disease. It is also very much large-scale impacts, and as you can see that a large part of the country is affected in the dry season by salinity intrusion and by flood. So the yellow and the red are salinity affected areas, and the blue areas are flood affected areas, and both areas are affected by cholera in spring and in fall, just like we saw in the previous slide. But here comes the interesting part.
Hello, I'm joining the call.
Hello, is this Dr. Colwell?
Yes,
Welcome to the press conference.
thank you.
And is Anwar Haq on the line too?
I don't know. I'm calling from Dublin, Ireland.
Yes, thank you so much for doing so. Well, we have one of the talks has begun already by Ali Akanda. Do you have time that you can sit through him finishing his talk and then you can speak, or would you like? Are you too limited on time, or would you like to take over?
Oh, I'm afraid that it's 11:15. I have a morning flight, and I'm also, it turns out that the conference call is international rates, so it would be very expensive to be on for an hour.
Okay, so why don't you go ahead and please give us whatever kind of talk you can, and then we will move on with the rest of the speakers.
Okay.
Thank you.
The points that I would like to make is that the work that we're doing allows us to develop eventually a predictive model for cholera, and that the work that Dr. Jutla has done is very helpful in correlating environmental parameters with epidemics. The work has separated, has shown a differentiation, I should say, between epidemic and endemic cholera. This is extremely important because epidemic cholera is that which occurs over a long period of time without cholera and with a sudden epidemic, and has been not understood as to why that happens when we know the bacterium is naturally occurring, is present in the environment, is part of the natural aquatic flora of the environment. And the concatenation of extreme weather, very warm temperatures, a very, very heavy, unusual rainfall, coupled with conditions of poor sanitation, lack of access to safe water, and crowded conditions, either refugee camps or some unusual gathering, religious festival or other gathering of a large number of people that exacerbate the conditions of poor sanitation. And the findings for a number of locations and outbreaks show that this model that we have proposed holds up rather well in a number of cases. So this work is a culmination, really, of about 40 years of doing research on cholera in Bangladesh, in the United States, in Chesapeake Bay, in the Gulf of Mexico, and in Haiti, and more recently in Africa. The genomics we have published as well, showing the presence of Vibrio cholera, particularly in Bangladesh, in various ponds with genomic differences, so that we have evidence that the outbreaks that occur are not necessarily a single clonal strain. So I think this is a paradigm shift in our thinking about, in our interpretation of cholera epidemiological data, and for the first time, hydrological, climatological data being incorporated in understanding the outbreaks. So I think these are the points that I would make very strongly. And I know that Antar, Dr. Jutla, can provide excellent description of the hydrological conditions and the downloading of data from satellite sensors. So I think the combination that we've been able to provide of molecular genetic genomic data, ecological data with respect to the distribution of the bacterium in the environment, and coupled with climate data, to the point where we even sort of extend ourselves a little bit, but I think it's worth hypothesizing what might happen with what is predicted to occur with climate change. So these are my points, and I'd be happy to answer any questions.
Okay, do we have any questions from the press in the room? Okay, hold on a sec.
Hi, my name's David Appel. I'm with Yale Climate Connections. I was wondering if you could have done a linear regression test to find out how which variables are most important, or how much they're important by, and things like that.
Yes, we have published papers on what we call ground truth data, where we have collected data on a very large number of parameters: conductivity, presence of heavy metals, various nutrients, nutrient concentrations, salinity, temperature, and salinity, of course, is a major factor, but I think there is a satellite now that can measure salinity, but that has not been available. From the oceanographic work and the estuary studies that we have done in the Bay of Bengal and Chesapeake Bay in the Gulf of Mexico, we have found that it is really temperature and sea surface height, which is related really to water depth, which Dr. Jutla has been able to refine in the analysis, so that the differentiation of spring and fall outbreaks has been able to be related to the depth of the river, or drought conditions, if you will. So the determination of the accuracy, or let's say the relevance of temperature and rainfall have been developed over the last 30 years. The actual statistical analysis, I'll leave that to Dr. Jutla to expand on with respect to the slides that he's presenting.
Do we have any other questions from here in the room? Do we have any questions from, do we have a question here? And please identify yourself and affiliation, thank you.
Okay. Kiki Sanford, This Week in Science and Boing Boing. I'm wondering, since you are developing these predictive models, is there anything that you are predicting currently for the next 10 years, or any specific time frames?
Well, the climatologists indicate that we can expect warming. And one note about warming that has already been published that we've worked on, which is quite fascinating, is that we have done a study with Dr. Carla Pruzzo from the University of Genoa and with the team at Southampton in England at the Oceanographic Station, where they have stored plankton samples over the last 40 years, and also data on warming of the ocean temperatures where the plankton samples have been collected. We've extracted DNA from the plankton samples, and we have probed them for vibrios, and we have found that indeed, along with the increase in sea temperature, even though very slight, there's been a consistent increase year by year relative to temperature of the numbers of Vibrios. And then a group in, I think, Denmark has done over that time analysis over that same time period of the cases of Vibrio infections, and indeed the numbers have gone up. In addition, for the first time a few years ago and subsequently since, in Alaska, Vibrio parahemolyticus, and a relative of Vibrio cholera, different species causing a different kind of infection, a foodborne infection, cases have been occurring with the warming in the Gulf of Alaska. So we have also ancillary evidence that these climate conditions really do have an effect on the native flora, which Vibrio is a component. So the predictions are that there will be increased temperature, ocean temperature warming, and there will be more severe storms. So one can anticipate that that combination in parts of the world where there's very poor sanitation, that the cholera outbreaks will be more frequent according to the conditions that institute them, or initiate them, I should say. And probably will be more intensive because the numbers of Vibrios would probably be higher. And we know from experiments done 50 years ago by other investigators doing volunteer studies that Vibrio is a dose-dependent disease. Cholera is dose-dependent. It takes about 1 million cells per milliliter for a full-blown case to be initiated, whereas a few Vibrios will cause a diarrhea, but not necessarily a severe, debilitating, and life-threatening diarrhea and vomiting, dehydration. So the evidence is accumulating in a very fascinating way.
Okay, we have another questions, which we will get to in just a second. And please identify yourself and your affiliation.
Laura Popik from Live Science. So you say this is a culmination of about 40 years of work, so I'm just wondering what your most immediate next step is in your research.
Well, I think it's really important to understand the genomics and the question of why it's so difficult to isolate toxigenic Vibrio cholerae from the environment, even though we can, with gene probes, detect the presence of the toxin genes and the presence also of the serotype genes. We know that the serotype genes are laterally transferred, and we know that the genes conferring toxigenicity are transmitted via a bacterial virus, a bacteriophage. So there's a very dynamic system in the environment. We know the Vibrio can laterally transfer about 80% of its total gene component. I mean, not all at once, but these genes can individually be transferred. So this is a highly versatile, mobile bacterium. And the question is whether the toxin is, in fact, perhaps a osmoregulator in its natural host, and therefore causes severe dehydration and fluid loss because of its effect in the human intestine. So these are the questions now, getting down to the actual interaction of the human and the bacterium and its role in the environment. So we have a lot of evidence, we have a lot of correlation, but it would be really nice to do the final putting together of all of the data into the full cycle of human, natural host in the environment, and climate.
Any questions from the chat? Any additional questions here in the room? Okay. Dr. Cobal, we sent you another email inviting you to watch the rest of the press conference on the web stream if you'd like, and you'd be also welcome. You'd be also welcome to register for it. Takes for just a second, and then log into the chat, so you could also still be available without having to pay international charges to participate.
Okay, but it is getting close to midnight here in Ireland, and I do have an early flight, so I may go on, but just for a few minutes.
Okay, well, thank you very, very much for joining us for the time you could, and we hope you'll participate a little longer if that's possible, and we'll move on with the rest of the press conference now. Thank you.
Thank you very much. Bye-bye.
I won't take more time. I will give Dr. Jitla some time to finish his slides. I will just mention that these two slides shows drastic increase in cholera over the last 30 years. We have put together over 30 years of prevalence, cholera incidence and prevalence data, which have not been worked on before. And this shows that prevalence has almost doubled over the last 3 decades. And you see the last 10 years have shown a very increased rate of growth. And if you divide them up in 3 decades, you see a very strong pattern of increase. And prevention is clearly failing, falling behind. And that's why we need early warnings, which Dr. Jitla will focus on.
Okay, so good afternoon. I have made 20 slides. I was under the impression I was going to talk for 20 minutes, but I'll speak for 5 minutes. And then as we go along during questions, answers, I'll try to cover up everything that I have prepared. So following what Dr. Caldwell has been discussing about epidemic and endemic cholera, what we have done is, so let me give you the results first. So for the epidemic cholera, meaning the cholera that occurs more suddenly, sporadically, and at an unknown location, these conditions are predictable at least 1 to 2 months in advance. For endemic cholera, meaning cholera in the locations where you see constant presence of cases all year round, these conditions are predictable at least 2 to 6 months in advance. Now, this is done with the help of satellites. And this is why the presentation was called Eyes in the Sky. The next key outcome is the changing climate. So for epidemic cholera, what we are seeing is new and unexpected regions, particularly in Asia and Africa, are likely to experience this form of disease outbreaks. For endemic cholera, we expect, at least from our modeling simulations, that there might be changes in the seasonality. Now, in order to understand these things, we need to emphasize that cholera can only occur if we have hydrology, climate, coupled with societal conditions. If there are no societal conditions that are conducive for diseases, cholera will not occur. Like we don't see cholera in the United States, but we do see cholera in parts of Africa, Asia, Latin America. And there has to be a convergence at appropriate temporal and spatial scales. Only then we can see a disease outbreak. So hydroclimatic is a jargon, so it basically means hydrology and climate. And if we can identify conditions which can provide the lead time, we can identify the hydrology and climatic conditions, it will give us a lead time to design the appropriate intervention and mitigation strategies. Okay. So just to give you a pictorial version of what Dr. Caldwell said, here is the distinction between epidemic and endemic cholera. Epidemic cholera is mostly observed in the inland locations, which is about 100 to 150 miles off coast. Endemic cholera, on the other hand, this cholera is mostly seen along coastal regions. And here, what you see is you see an outbreak, but there are constant presence of cases. The disease is present in that region for the entire year round. But you will see a sudden outbreaks at a particular time of the year. Now, the challenge for hydrologists or geoscientists were to identify if there are any hydroclimatic conditions that can tell us or that can inform us on how these two kinds of cholera be quantified or be predicted. So our hypothesis for epidemic cholera is that if we have above average air temperature, which creates the warming scenarios, and it is followed by above average precipitation in combination with already debilitated or poor sanitation conditions, we are likely to see a cholera outbreak. So we tested this hypothesis in various locations, like here we have tested it over Central African Republic, we tested it over Rwanda, we tested it over Mozambique, and we are seeing these consistent patterns. Now, remember, the challenge is that these outbreaks, like epidemic outbreaks, are very hard to, the data is very hard to obtain, meaning that we don't know the timing and location of these outbreaks. So a traditional correlation analysis or a linear regression analysis may not be valid on this type of understanding. Okay. Now, we see the similar thing with Juba in South Sudan this May, where we actually predicted that there is a greater likelihood of a disease outbreak in May of 2014. And interestingly, in Haiti, and this is a published study, so in Haiti, we saw that the air temperatures were above average, and when it followed with above average precipitation, we did see a cholera outbreak in October, and that was an epidemic cholera. Okay. So these are like snapshot type of studies done in different parts of the globe, and in around like 20 to 25 locations, and all of them are presenting a very consistent picture. So let me go through this one when we do a question session, if at all. So let me skim through endemic cholera. So endemic cholera is more in the coastal regions. So as Dr. Cole and Dr. Akanda has mentioned before, that these types of cholera has usually like a very strong seasonal cycle. For example, in Bangladesh, we have 2 peaks, which is very specific to that region. So what happens is that this first peak is a water scarcity driven peak. This is when river discharge is low, and then there is a intrusion of bacteria. And this bacteria, the coasts are the environmental niches of bacteria. So the intrusion happens to the inland water bodies, and then you see the first peak. On the other hand, the second peak is basically the water abundance peak, meaning that it is mostly flood dominated peak in the endemic regions. Okay, so how do we predict it? So we brought in the satellites. So we have the GRACE satellite system, which measures the total water available in the basin. Now, these are 2 very big rivers in the Ganges, Brahmaputra, and Meghna basin. And the GRACE satellites will provide us, and they provide us the total water available in the basin. So this water is, this anomaly is strongly linked at a 2 to 3 month lead time with river discharge. And river discharge has a 2 to 3 month lead time with cholera outbreaks. So in total, there is about 4 to 6 months of lead time. Now, if we relate the total water storage in the basin with the cholera outbreak time series, and this is based on like 10 years because this is endemic cholera, we get about 5 to 6 months of lead time when we can know the hydroclimatic conditions which are favorable for this disease outbreak. Okay. The evidence is presented here, where you see the river discharge. The solid line is the observed river discharge. So solid is the simulated one. The dotted are the observed points for river discharge at Dhaka location. When we predicted these spring peaks, we see a satisfactory performance of our logistical model for cholera outbreaks. First for spring, for the first peak. A similar experiment done for the fall or the autumn peak, second peak, is also yielding satisfactory results in our statistical simulation models. And I'm going to take 2 or 3 more minutes before I'll finish it up. So up to now, we used a cluster of satellites. We used GRACE for endemic. We used TRIM for precipitation in Africa. We used land surface temperatures as temperature values from MODIS. So there are like cluster of satellites that we have used to develop the prediction or the simulation mechanisms. Now, based on this understanding, let us see what happens if under climate change scenarios. So again, I'm going to take 2 more minutes. So let us focus on the endemic cholera first. So this is a statistical simulation. So you see the 2 peaks in Bangladesh or in the Bengal Delta, one the spring outbreak, one the autumn outbreak. So this is the ensemble run for A1B scenarios of GFDL and ECHEM. What we are seeing is a strong shift in autumn outbreak seasonality with a dip in spring outbreaks. And if we see the river discharge seasonalities or river discharge patterns, they are very similar. We see that the high river discharge is actually showing a decreasing trend. And this is what has been observed by other scientists as well. And we are seeing an increase in the low flow conditions. And low flows are actually responsible for the spring peaks. Okay? But the more interesting thing is the epidemic cholera, because epidemic cholera has more mortalities. So what we did was we did a synthetic run based on our hypothesis of elevated temperature and precipitation. So on the left-hand side, you will see that this is the population increase map up to 2025. So the blue is where we expect more than 50% increase in population. So let's see what happens to epidemic cholera in Africa. So in these slides, the more the red is, the worse the cholera becomes. So this is for 2020s, 2030s, then we move on to 2040. So this is done over each decade. Okay, so you see that the entire Central Africa by 2050s is at a likelihood of increased epidemic cholera outbreaks. Okay, this trend or this analysis was actually done for GFDL model and the ECHEM models, and both models are showing us a very similar results. But in order to maintain the time, let's see what happens to cholera in Asia. So we did a similar analysis for epidemic cholera using GFDL model and A1B scenarios. So the red ones is the region where we are likely to see an increased cholera epidemics. So this is again done on decadal scales. So as time progresses, the likelihood of epidemic cholera in Asia, at least in Bengal Delta and in Mekong Basin, is going to increase. Again, this is a hydroclimatological simulation. It does not take into account the societal determinants into it. Okay. Then moving on to next to the epidemic cholera in Latin America or South America, we'll see that how this region, which is at a very low risk of cholera, becomes at a high risk region for this epidemics. Again, like I must caution you that this is a simulation result. This is a climate change model simulation result, so it does not have a societal component yet into it. Okay, and with this, I'll thank you for your time.
Hi, is this on? Hi, my name is Joanna Wendell. I'm from the American Geophysical Union's newspaper, EOS. You said that these models so far don't take into account the societal effects, but do you have plans to somehow include them, or?
Yes. So right now we are working on determining the appropriate societal conditions. We have plans to incorporate or to actually, so we have identified some conditions under which this cholera outbreak happens. One is the water insecurity, and one is the community resilience. So we are working with sociologists to incorporate that plan into our models. But as of now, the population is also increasing, is also playing a role. So we are incorporating the data, CDAC data, into our prediction modeling simulations. Actually, this entire scenarios, or this entire prediction mechanisms is now linked with Google Earth. So we plan to release it in a user-friendly type of mechanisms in like 6-7 months. So we have results to show you in a Google Earth as well, but this computer, I don't know how this computer
Just
will work.
one small point to add. One interesting thing about the simulations Dr. Jitla showed was that they are based on the A1B scenarios. So they have a certain set of development and energy emission scenarios already embedded in it. So they have some indirect input of societal conditions already. But of course, as he mentions, that a more accurate way would be to actually include direct data into it, the simulation models.
Jeff Hainstyles, Passport to Knowledge. Dr. Colwell and you both said things like early warnings are essential. You talked about lead time. My understanding is that one response to threats of cholera is to have pre-positioning of oral rehydration salts so that they're ready to go into the field, so that the local health authorities can essentially respond appropriately. What interest have you found from the governments of the regions that you're studying and or the World Health Organization to turn predictive models into operational realities on the ground?
Sure. In the endemic regions that I have worked on, the ICDDRB, the International Center for Diarrheal Disease Research, they are based in Bangladesh. They have shown great interest, and we are still working with a couple of their scientists. So they are probably best positioned at this point to use or take advantage of the type of work that we are doing. But of course, they're in the rapid march in the African countries, it's almost like a moving goalpost here that we have to work with many more authorities or countries in the African region. But as the most of the understanding comes from the endemic regions, and those countries are definitely already trying to make use of the information that or the scientific discoveries that we are working on.
And to add into what Dr. Khanda is saying, I think the best person would be to have this answer from Dr. Caldwell as well, because she travels a lot and she has more connections with WHO. So I would correct.
Sure.
Madeline Nash, freelance. Do you expect any change or spread of the endemic regions of cholera? In other words, do you expect cholera to establish itself endemically around South America, around Africa, these new places where you're picking up epidemic cholera?
Well, cholera bacteria is native to coastal environments, and the frequency or the growth conditions may or may not be conducive for bacteria to survive. For example, if we have certain above threshold air temperatures, then water temperatures, cholera bacteria may not survive or may survive. But this is a point of uncertainty in our models as of now. So it's not yet clear to us if the endemic cholera will become more or less in Latin America. But we are progressing. We are working towards that.
David Appel with Yale Climate Connections. What about China? Is there no good data on China?
Well, I haven't seen, at least I haven't seen literature that has data on China. But again, like the call may have some better insights.
What about post-war areas where there's a war, where they've had a significant war, especially in the Middle East? Are they prone to cholera outbreaks?
Any societal condition that devastates the water and sanitation infrastructure puts communities at risk. But we need to have appropriate hydroclimatological conditions like certain temperatures and precipitation to create the transmission mechanism so that trigger of cholera can occur. So there is an instance that in 2007 or 8 there was a cholera outbreak in Kirkuk region in Iraq. We have not looked into the hydroclimatic conditions yet, but there was a mild outbreak reported, at least in WHO databases.
Laura Popik with Life Science. So is there another reason that there are outbreaks due to low water conditions, aside from the fact that they're just higher quantities, or there's a higher density of the bacterium, or is there another reason why those conditions induce outbreaks and low river conditions?
Okay, so you mean the spring cholera probably that we were discussing. So there can be potentially two explanations for that. One is that, as you mentioned, that during low water conditions there might be higher concentrations of the pathogen, so it makes more people vulnerable and more people tend to converge on few compromised sources. But as Dr. Jitla also showed, that in that season, due to less freshwater availability from upstream, there is a tendency of coastal intrusion in delta areas. So this coastal intrusion includes salinity as well as bacterial concentration in the coastal areas, because salinity is a great, it helps growth of E. cholera and Vibrio cholerae. And so the extent of these coastal intrusion as well as the magnitude of these low flow conditions, that can increase a larger region, that can make a larger region vulnerable to these outbreaks, because your water resources are compromised. At the same time, there is a higher concentration of the pathogen in the regional rivers or estuarine rivers. So that could be a secondary reason how more people could get affected.
So we have time for one last question.
Are there any future satellite missions planned that will continue the work or help improve or help?
I guess GPM,
Yeah.
and SMAP is also helpful, is likely to be helpful, but again, like these are speculative answers right now.
So sensors that would help us determine extreme precipitation events or soil moisture conditions or better river discharge conditions would be definitely helpful. So those are the ones.
Yeah, and then better resolutions, perhaps, spatial resolution. That's it.
Okay, I'd like to mention that there's a statement, a printed statement from Dr. Colwell on the table in the back, if you want to pick one up. Also, as the day progresses, we're posting slides of people who have given talks in press conferences and supplementary material on the virtual press room on the AGU Fall Meeting website. Click on the media button, it'll take you to the media center. You'll see in there a widget that says virtual press room, and you can download press releases and stuff from the speakers there. And thank you very much for your talks. I'm glad we were able to include Dr. Colwell.
Yes.
And thanks everyone else for being here and asking questions. I also wanted to just say that we have one last press conference today. That will be at 5 o'clock, and that one is about new insights into boulder transport by major storms.